So I asked you guys a quick question. Get reconvened. The three basic types of rock. I know there's more than three. Let's see what you came up with. Because we're going to be talking about fossils today. Oh yes, the three: alternative, heavy, and classical. Yes. Igneous, metamorphic, and sedimentary. Perfect. Modern, soft, and hard rock. Yeah. Boo. Can you tell it's Halloween? Right. Yes. Liquid, solid, gold.
You're welcome.
Solid gas plasma? No, not. Oh, geez. Yes. Kane, I'm gonna send this YouTube link to your parents. All right. Oh. All right. I don't know. I'm not comfortable with that. Yeah. What? I do. What? Oh, yes. So we're here to talk a little bit about fossils, which obviously are going to involve different layers of rock. But we know fossils are the remains and traces of past life, and we understand that paleontology is that study of the fossil record. We all have probably taken a look at dinosaurs a little too much in our history or elementary years, right? And probably not paid too much attention to anything else. So hopefully we get to look at some of those other periods and eras that are a little more important than our dinosaurs. Because believe it or not, dinosaurs, just like us, are kind of a small blurb in the history of Earth. A very cool one, you know. Yes, dinosaurs are everything. I love looking at dinosaur fossils. They are very cool, but there are much older fossils right under our own feet here in Michigan that wash up on shore. Our state stone is one of those, right? Our state rock. Hexagaria is the coral that makes the Petoskey stone. If you ever look closely at a Petoskey stone, it has 6 sides, right? It's a 6-sided coral structure. So Michigan is, I like to say, a lot more interesting than like Utah and places they find all these big fossils, these dinosaur fossils, because we have older ones, way more prehistoric. But sediment, we know, is the type of rock that embeds these fossils and allows fossils to be created and made and for us to find millions of years later. And sediment is produced by weathering and erosion of rocks, and sediment becomes a recognizable stratum. And if you guys remember all of those layers of Earth, each one is called a strata. Together they make the stratum, okay, in the stratigraphic sequence. So when we look at stratigraphy. I can never get those 2 R's out. I was doing that with my daughter the other day. It was funny because we were looking at different stratums and layers, and I tried to say it, the study of it. I couldn't say it then. I still can't now. I feel dumb. I just said that on YouTube. I do. I feel like I must have sucked too many dum-dums this morning, right? All right, so the stratum of the same age tend to contain the same or similar fossil assemblages, and this includes index fossils. Now, if I get to a layer of Earth and I see that I'm finding a common fossil there, and it's in that particular stratum all over the place, I then refer to it as an index fossil. Now, we know all of the continents used to be together, correct? Okay, Pangaea, whatever you want to, whichever stage of the plate tectonic shifting you want to stop our continents in, you can have many different continents, we'll say, or just one. So you guys, at one time they were all together. If you put your fingers next to each other and then they shift apart and some become higher, lower, break apart, it doesn't matter because all those buried layers should be the same in all of those edges, so to speak, or the continental plates, okay, or the oceanic plates, I should say. So this helps geologists determine relative dates of embedded fossils despite upheavals, and we refer to that as relative dating. So now, when I say relative dating, I'm not talking about the royal family, you know. I had to tell the joke. Okay, so I'm. Okay, so oh, you did. Sorry. Okay. That's why they're all extinct, right? Because there's another one. Okay, so you guys, I really want you to think about this because this is important. If I have all these buried layers, and then I can radiometrically date each layer, and I know there's a certain type of fossil in each layer, if I find a layer on a different continent that we know was around or together at one time, do I need to radiometrically date it when I find that particular fossil in a stratum, or do I already know it now? You know it, right? Okay? And because we know it, we don't have to do radiometric dating all the time. Then we can use something called relative dating, which is less accurate, but not really, because we've already done all the radiometric testing to relatively date things. So really, you can't have one without the other. What questions do you guys have about either of those types of dating? Because if you aren't clear on radiometric dating, that's called, that's like how you absolutely date something. That's what it's called, absolute dating. Or based on relative dating, what does that mean? What questions do you have about those two terms, if any? If you don't have any, that's awesome. I'm scared to see what's going to come on my screen, to tell you the truth, after the jokes. Do you have any questions? Oh, no. Nope. Don't. Okay. 13 responses. Where are they all at? Uh-oh. I might be getting froze up on Pear Deck. I'm getting a lot of lag here. Let's go ahead and move on. I don't know what's going on there. So what you're seeing here on the side of this rock is many different stratums. Has anybody been to a giant quarry? Yeah? If you drive, anybody ever drove to Wisconsin? Yeah? Have you seen the quarries along the way there? Okay. Like through Milwaukee? Okay. And so, and in Michigan, but I mean, there's some really huge ones. And what you're seeing as you drive even through the mountains, you look at the side and you're like, geez, they cut that rock in a really weird linear way or horizontal way. Why would they do that? The truth is they didn't, right? They just broke the rock off, and you can see the stratum layers right in front of you, okay, caused by all the sedimentary rock being formed time after time, okay? So these are the layers. Now let me explain that. That means that this layer, let's pretend this layer is old enough to be in all continents, would be the same in our continent, North America, as it would be, let's say, in Europe or in Africa. All of those layers would have a similar fossil in them. So let's say it is let's just say it's Hexagarya, our state stone, in this layer. That means no matter where I go, if I dig down to that layer on any continent, I should find that Hexagarya coral. Okay? That would be an index fossil. So here we are talking about fossils. If I ask you the question, what type of rock, now that I've asked you about the three types of rock, makes fossils? What type of rock makes fossils? This should be a rather simple question to answer. I don't have that number memorized, but I can figure it out for you. It's not very fast. It's about the same rate at which we make soil, which is like 1 millimeter or 2 a year. So, right? It's Halloween. You guys can have a little fun. Let's see if they're actually coming through today. Yeah, sedimentary rock. Alternative rocks. Okay. Yeah. I saw all King's Ave. Oh dear. That is. I like it. I like it. Yes. Is anybody dressing up as Sherlock Holmes tonight? No. The new Elementary was on. I did see that. I recorded it. I haven't seen it. What? Yeah, I like entertaining things sometimes. Like once on the weekend or twice. Yeah. I record some Discovery stuff too. My wife makes me watch like the originals and the Vampire Diaries though, so I'm not. I kind of like it secretively. I pretend like I don't, but I'm entertained by it. All right, so we talked about relative dating, looking at layers of the Earth, and then I mentioned absolute dating, and this assigns an actual date to a fossil. Now, often in earth science, they talk about these two things like you do one or the other, and then your earth science or geology teacher will ask you which one's better. And the truth of the matter is geologists don't use just one. They use both. Okay? And one really can't stand alone without the other. Now, one absolute dating method relies on what's called radiometric dating. You guys know one version of radiometric dating by heart. I'm talking about carbon dating, right? But remember, there are 118 elements on the periodic table that's rolled up behind me, okay? And all of them have isotopes, and all of those isotopes have half-lives, and I can use any one of those isotopes that I find in the Earth to radiometrically date what I'm looking at, okay? So that refers to half-life. Now, this is the length of time that is required for half of the atoms to change into another stable element. And we did a lab over that, right, the dinosaur dig? Okay, all right. So unaffected by temperature, light, pressure. I can't do that lab in class. I don't have the $1 million machine to radiometrically date things and measure isotopes, but it would be cool if I did, right? Now, all radioactive isotopes do have a dependable half-life. Now, this is always the area of contention for some people because carbon dating is really only good for like up to 10,000 years or so. Some say 30,000. So is it usually carbon that we're using to date things? No. Okay, but it is what people use to discredit carbon dating. And I said, well, you don't really know your chemistry very well because if you look at real radiometric dating, they're using a multitude of isotopes when they radiometrically date fossils. And they usually aren't dating the fossil themselves, are they? They're dating the material that's around the fossil, right? Okay. So some billions of years ago and most in between. So some of them we can date back to 4 billion years. That's how we know how old the Earth is. Some are in millions of years. Some are in a few seconds, okay, literally. They break down that quickly. Now, many isotopes are used, and then they combine their half-lives and they make them useful over all periods of interest. So depending on how deep I dig in the stratum will depend on which isotope I'm going to use. So if I tell you something, I'm going to give you a little problem here. I'll write it out here, and then I'll let you guys answer it. Oops. So if I tell you something has a half-life of, let's say, 25 years, okay? Let's go 25 million years. That looks like 1.5. You know what I mean, though. 1 half-life. Okay? I'll put a dash in there. Okay? So 25 million years is 1 half-life. And let's say I have, 25% of my parent isotopes left. Yeah. Is somebody missing their life? Oh, okay. Thank you very much. He owes you his life. All right, so I want you to tell me how many half-lives have passed for this particular element, whatever it might be. If 25% of my parent isotopes are left, how many half-lives have passed? Better yet, tell me how many half-lives and how many years. If there's 25% parent isotopes, that means that there's 75% daughter isotopes left, right? And do not tell me all atoms are isotopes. I know that. I'm so sick of hearing that. Yeah, but here's the thing. We wouldn't have the word isotope if we didn't refer to them as things that have a different number of neutrons and protons. So they are isotopes in biology, we'll say. Okay. In chemistry, everything's an isotope, right? I get it. Let's see what you guys came up with. Getting some really, okay, it is 2 half-lives, and it should be 50 million years. Let me explain that. I'll go back to the problem real quick. Because, believe it or not, this is one of the hardest things for some reason for kids to get. If I start off with 100% of being 100% parent isotopes, like the pure isotope that I start with, okay, and I only have 25% left, that means it's been cut in half twice, right? 100% to 50%, 50% to 25%, that's 2 half-lives. And if 2 half-lives our present, we know that 50 million years must have passed because each half-life took 25 million years. Now, depending on what your half-life is and which isotope you're using, that will change depending on which fossil or stratum you're looking at. Okay? All right. So the geologic time scale, that's where this whole time scale comes from. That's how we can say dinosaurs and humans never were around together, because I cannot talk today. I have no idea. It must be Friday. All right. My words have been coming out of my mouth conjumbled all morning. Something's happening. Yes. So, yes, they squeak. So, something's going on. I'm just thinking about all that candy, and then my kids are going to be stressing me out all night. I know. You guys wait. Someday. They like stay up till 1 and just squeal and scream on a sugar buzz, and then all of a sudden they drop dead at 12:30. And it's like, oh, they're down. Thank God. All right, so we have to give a layer or a date to each layer of the Earth. Without those dates, the layers of the Earth mean nothing. This is how we know that humans and dinosaurs did not exist together. There is literally 65 million years of rock between us, like the very first modern human, and dinosaurs. And what's going on? Is that James Taylor? Okay. Oh, it just got bumped. What was that? Oh, was that Mr. B's music? Okay. All right. I'm not dumb. I'll go with it. Okay, Mr. B's music. It was bad enough, right? I'm teasing. It wasn't like teeny bopper music from the 80s that he usually plays. So I knew it wasn't his. Was it James Taylor? Is that what that was? I couldn't tell. No? I don't know. All right, guys. I know I had a thought, and now I've lost it. So that's okay. So looking at us and dinosaurs, you guys, there's way too many layers of the Earth between us and dinosaurs. There's no way we would have been around together. So we're going to divide them into eras, periods, and epochs, and this derives from the accumulation of data from the age of fossils and strata all over the world, and life arose during what's called the Precambrian time. Let me ask you this question. Why are there not very many fossils from the Precambrian time? Why are there not many fossils from Precambrian time? I do somewhere. Let's see what you guys came up with. This is the other debate I have with people all the time. The Cambrian explosion. And I say, it's not really an explosion. Not really. Kind of. No bones? Okay, that's true. No bones at all. Okay, no skeleton is the best answer there, right? Couldn't be fossilized. We're talking all of these mushy, soft tissue creatures, right? Okay? Like, think of jellyfish. Exoskeleton. What is that? Skeleton. Exoskeleton.
No. It wasn't enough of that. Apparently there's no at the top.
Okay. Lots of single-celled organisms, and not just that, we are lacking a skeletal structure. So imagine this. You have to bury things in sediment to get a fossil. Okay? And they have to hold a shape in that buried pile. Okay? Even algae have chitinous material, right? And so it has to have some type of skeleton. And then after Precambrian time, of course, comes the Cambrian explosion. And the biggest question that I've had to answer to a lot of people, because they always ask me this, well, what about the Cambrian explosion? How do you explain that? And I said, well, it really wasn't an explosion because life had been evolving in this very diverse way for a long period of time. It's just that was the onset of more hard exoskeletons, right? And it wasn't really an explosion. I know it sounds kind of weird to say that because we refer to it as that, but it was just new developments and new abilities of organisms started to show up. And I did put this in your notes. We're going to go over all of these, but I'm going to leave that here as just a reference in your notes. This is in your notes, isn't it? It's in your textbook. Don't worry about it. I thought I put it in your notes. Well, I might make a copy of it and give it to you just so you have one to reference. So we're going to go through each different major era and period. So looking at the Precambrian time, this includes about 87% of the geological time scale. That means it's a huge portion of life. Why? Because that's when life started. That's when the most different varieties of life would have existed. Can I prove that? No. Okay. The reason you can't prove that is because we lack the fossil record. So little or no atmospheric oxygen in the early atmosphere would have existed, and the lack of ozone shield allowed UV radiation to bombard Earth. And the only place you would have found life starting to form would have, again, been in the very depths of the ocean. Now, the first cells came into existence in aquatic environments. Of course, here we are talking about the development of prokaryotes, single-celled bacterial-type organisms. And then developing into cyanobacteria. Do you guys remember what cyanobacteria do? Just yell it out. Photosynthesis. They're photosynthesizing bacteria. You should know that. Okay. Cyanobacteria are these photosynthetic bacteria. Okay. And the onset of cyanobacteria is what started to change our atmosphere from high in CO2 to high in oxygen. And we find these in early stromatolites. Those are like these protrusion rocks that will come up through the shorelines. I think there's some pictures of them. I'll show you some in a minute. But photosynthetic cyanobacteria added oxygen to the atmosphere, which allowed aerobic respiration to evolve. And once that happened, we started to get bacteria that went through aerobic respiration in that oxygen-rich atmosphere, which sounds great, except oxygen likes to do what to things? Oxidize them, right? Okay. So it's like a double-edged sword. You want this perfect combination of CO2 and oxygen in your atmosphere, and most of the air we breathe in is what? Nitrogen, right? N2. Like almost 80% of what you breathe in is nitrogen. So you don't want too much oxygen. It would literally oxidize you. It would be terrible. You wouldn't want that much. A little bit of it's good for aerobic respiration. Too much would be bad. So looking at the tree of life and what we were just talking about, you start off at those early first cells developing into prokaryotes and bacteria. And I want to talk about the two types of bacteria. We'll spend more time on this later. But the archaebacteria, archae is referencing our archaic ancestors. Okay, so these are the bacteria that we're related to as eukaryotes. And these other bacteria are kind of the bacteria that we can still find today and that we refer to as actually being bacteria. And then we have this other kind of bacteria. These are the ones that get us sick and things like that. And these are called the eubacteria, okay? Eubacteria, right? Okay. And that's where we would have evolved from as well, like animals, fungi, protists, and plants all came from that ancient archaebacteria. And we still find some today in their original states. We find them as extremophiles in volcanic vents, deep geothermal, hydrothermal vents in the ocean. We still find these things that can live in like acid, basically. Okay, and we call them extremophiles. And they are the remnants of our early archaea. Because think about Earth's early atmosphere and early planet. It would have been a very caustic place, very caustic. The only thing that would have been able to live there would have been these thermophiles, halophiles, and methanogens. Now, eukaryotic cells arose about 2.21 billion years ago, a long time ago. And most are aerobic, and they contain a nucleus as well as other membranous organelles. Now, we do have some fossil evidence of this. They have found these cyanobacteria balls that a lot of them are fossilized. They're just piles of this stuff. And if you go to museums, I think I mentioned this, you'll see this giant rock with a sign next to it. A lot of times that is a fossilized pile of early either zooplankton or phytoplankton that contains a lot of these single-celled organisms. Now the endosymbiotic theory, does anybody, let's see if you guys remember that one. What is the endosymbiotic theory? Tell me. If you don't remember, it's okay. Just say, I don't remember. IDR. I don't remember. Oh, close. Je ne comprends pas. Okay. Je ne sais pas. Okay. Tu es stupide. That's me. That was mean. I don't know if it. I don't know. I know. I remember a little. I took through French 2 in college. I only remember a little bit. Okay. So I'd say about 10% of you kind of got it right. So inside of our cells, and we'll talk more about this later in the year, we have mitochondrion, right? Now, mitochondrion are kind of a creepy little creature that's inside of us. It sounds weird to think of them as their own organism, but in a way they are. They actually have bacterial DNA. They actually have a bacterial structure. They have a double membrane, okay? And they have these two layers, almost like a capsulated bacteria inside of us. They're the same size as the bacteria. Guess what? They have their own ribosomes that make their own proteins. And we don't get half of our mitochondria from our dad and half of it from our mom. We get it from our mom like an infection very early in development. Now, it's a very good infection, right? I mean, it gives us energy. It gives us ATP. And without it, we don't exist. Chloroplasts are very similar. They have bacterial DNA. They have their own ribosomes to make their own proteins. So where did these things come from? Well, it's thought that an earlier, larger prokaryote engulfed either these mitochondrion or chloroplasts, and instead of them destroying each other, they formed a relationship, kind of like an orchid and an insect that would pollinate it. And from that, we get the endosymbiotic theory. The idea that just like a micelle would have to engulf an enzyme, another prokaryote would have had to engulf another prokaryote, and they would have not had to kill each other, right, in order to get multicellular life. That's the only way we get to something that has enough energy to have more than one cell, okay? And that is the endosymbiotic theory. If you've had me, you know that, but I just want to make sure we talked about that for a second. Some, right? Some. They actually trade genes, right? Transposons, plasmids. So mitochondria were probably once free-living aerobic prokaryotes. I say aerobic because they go through what? Respiration, right? Okay. This is where oxygen combines with hydrogen to go kablooey. Instead of kablooey, though, it captures it in ATP. We'll talk about that later. And chloroplasts were probably once free-living photosynthetic prokaryotes as well. And then a nucleated cell probably engulfed these prokaryotes that became various organelles. And then we start to really think about fat bubbles, right? Really think about bubbles inside of bubbles. You guys were having a talk about universes inside of universes. So let's go way down. Let's just think about our bodies, right? We're a giant set of systems together. We break those systems down into these other encapsulated bubbles called organs that do all different sorts of things. Inside of those organs, we have tissues, which are layers of bubbles, of different types of bubbles called cells, right? Inside of the cells, we have even more of these bubbles, okay? And I look at a mitochondria and I say, well, that's just a phospholipid layer that has a specialized function with all these cool proteins kind of arranged in it that help it make ATP synthases in there, right? Drives the whole process. And then you start to think about the endoplasmic reticulum and all the vesicles that are being made. And then you think about the nucleus, which is a fat bubble, right, with holes in it and pores that allow things to pass through it like RNA. And so really all we're talking about is all these different micelle and phospholipid layers all put together into a cell, and then more of those into tissues. And that's the way that evolution, early evolution, would have happened. And our cells are no different. Okay? We need to have these different organelles, and they would have had to evolve just like any organism has to evolve through natural selection. So cilia and flagella, you guys remember what flagella are? Okay, the tails, or you can be multiflagellated too. Okay? They may have originated from the slender, undulating prokaryotes that attach to the host cell. So we still have these today. Some E. coli are flagellated. They can swim upstream. That's not good if you have a catheter in, right? You can get a urinary tract infection. That happens quite a bit in hospitals, by the way. The bacteria, they can swim upstream from a catheter that comes through the urethra. Because if somebody's having problems going to the bathroom, they cath them. Basically, they pipe their urinary tract. It looks painful. I don't know. I've never had it done. But if they don't change that every day, the bacteria will swim up it, and because the bottom is contaminated, obviously, they'll actually swim up that catheter and they'll get a urinary tract infection from that. Okay? It's kind of gross when you think about it. It's like you got these little creatures swimming upstream. All right. So support for the endosymbiotic theory is mitochondria and chloroplasts are similar in size to bacteria. They both have their own DNA and make some of their own proteins. And yes, they have their own ribosomes. And mitochondria and chloroplasts, they don't divide like your cell. They actually go through binary fission, just like a bacteria, exactly the same way, exactly the same time, which is really creepy if you think about it. And the outer membrane of mitochondria and chloroplasts are different. The outer membrane resembles a eukaryotic membrane, but the inner membrane resembles a prokaryotic membrane. So the membrane that surrounds the core of what a mitochondria and chloroplast are is just like a bacterial one. And I always think of this as kind of the coolest thing that we learn in biology in a lot of ways, because it is why we're here. It's why all animals are here. It's why a plant is here. It's why anything more than one cell is able to happen. Now, multicellularity arises about 1.4 billion years ago. So all of a sudden, not all of a sudden, I guess billions of years have passed, right? But we start to develop the ability to put many of these respirating cells together and photosynthesizing cells. And this separating germ cells from somatic cells may have contributed to the diversity of organisms, having the ability to have sexes. Or asex, right? Think about this. Females came way before men, way before. And I'm not talking about humans. I'm talking about just in general. The female would have had to come way before the male. That's why everything in biology is called mother or daughter, right? And so we take a look at our germ cells. I would have had a female germ cell first. Now I want you to think about an X and a Y chromosome. Let me just draw one here. We did this the other day in biotech. How about this? I'm going to ask you, which one of these would be representative of a X chromosome? And my pen's not working. Come on. A, we'll just do that. So A would be X, B would be Y. Why? There's not as much genetic information on the male chromosome because it's not been around that long, right? It hasn't had enough time to evolve and change and develop new genes. It's that simple, okay? And infinitely, females are way more complicated than males, right? That just goes without saying, doesn't it? A little bit, okay? I've never seen. Never mind. I'm gonna get in trouble. No. I just know when I pick out shoes, it's done in like 5 minutes. Like, I want those. I'm good. Don't you want to look around? No. Those. Right there. All right. So early multicellular organisms lacked internal organs and could have absorbed nutrients from the sea, and it's possible that practice sexual reproduction could have started there. Now, I will say, it's not like the male gender or males in general are insignificant. They do one major thing for species. They allow us to adapt. and change and have much and many varieties of who we are. They mix up the gene pool, okay, which is extremely important if you want to survive an ever-changing environment. You need to have lots of variety. So we are good for something, guys. And the human species has evolved too far to have just females, right? There's no parthenogenesis in humans. There is in frogs, though, right? You don't need a boy frog. Girls can self-fertilize. It's kind of weird, right? A lot of reptiles can do that too. Do you know you can change the alligator sex of an egg just by changing the temperature? I believe if it gets too hot, they become
males really like it. It gets cold or too hot.
Yeah, I can't remember which one it is. I think if it gets too hot, they become male. Like literally, you can change the sex, even if it has the chromosome. It doesn't matter. Their sex chromosomes are different than ours, so they're less evolved. Ours are more specialized. Humans can't do that, obviously. It is weird, right? Here's another thing, and I'm gonna put this on YouTube. I don't know if I should even talk about it, but if male testicles don't descend during development, they actually can become ovaries, which is scary in itself, right? Okay. It's all a temperature thing. It really is. So. No.
What you talk about on YouTube?
Oh yeah, I know, right?
No.
It's related. It's all yeah, I know. So the nice thing about biology is you can answer all these fun questions and mysteries sometimes, not all of them. So the next one is the Paleozoic era, and this begins with the Cambrian period, and this lasted over 300 million years. This includes 3 major mass extinction events, and this is where the disappearance of a large number of taxa or taxonomic species came in, and this occurred within a relatively short time interval compared to that overall geologic scale. We say quick, a million years is quick. It's really hard when you talk about geologic time for us because, we're only alive maybe 100 years, and so when you start talking even millions of years, it's tough. Even 1,000 years ago is tough for me to grasp. Even if you're the best historian on the planet, to fully understand even 5,000 years would be extremely tough, right? And now imagine millions. Whoa. Yeah, right? That's just cool. Yeah. So I think I'm going to stop here, okay? Because it's Halloween and I don't want to kill you guys today.